Okay, so the next panel is a very cool panel as well that will be moderated by Richard Soul. Richard, I'm not gonna steal your show. I'll let you introduce your panel and take it away.
You're on mute though.
That might help, right? Okay. So I want to thank the Children's Tumor Foundation for this opportunity to moderate the panel discussion today. I have I'm very honored and humbled by having a number of really outstanding researchers join me in this panel. Jonathan Chernoff from Fox Chase, Bob Van Huizenbroek, who's from UCL. He's a PI3K expert and will probably have more to say about the topic. Christina Fernandez-Val from University of Central Florida, and Dan Nomura from Berkeley. So thank you very much all for joining, and I appreciate it. So why don't we get into the topic of first of all, we just talk about back, and it might be worthwhile to talk about some of these mechanisms that are signaling mechanisms that are activated upon Merlin's loss of activity. And so why don't we go first to Jonathan? Why about what we're finding with FAC and PAC? Because I know you're working on PAC, so take it away.
Right. So when I think about this problem, and I've been working on it for quite some time, we know that the Merlin protein is the source of sort of aberrant signaling here because of its loss of function. But as mentioned earlier, that can be addressed perhaps by some kind of gene therapy, or in the rare cases of missense mutation, there might be a way to stabilize the conformation with a small molecule. I'm not going to really get into that. But 20 years ago, our group, Joe Kissel with Tyler Jacks, both found a convergence of Merlin with this PAC kinase that I had been working on sometime earlier. And that raised a question, would its relationship to Merlin be something like what RAF is to RAS? In other words, a key effector that you might be able to exploit for therapeutic purposes. And we knew that there was a very direct physical connection between these things. For example, PAC directly phosphorylates Merlin. Merlin binds to PAC. It's mutually inhibitory embrace that these two proteins have. So it had sort of a good characteristics as a potentially useful effector to go after. And, it wasn't just us finding that, but others as well. And I think importantly, Joe Kissel's group showed that in vivo from, samples from patients with schwannomas, endogenous PAC was in fact activated. So this wasn't some kind of interesting laboratory artifact, but something quite real. And so, small molecules became a thing to go after, and a lot of companies got involved with this. The most well-known, Pfizer had a compound, and then Afraxis, a small company, developed a series of inhibitors, and Genentech was in this game, AstraZeneca as well. And, those compounds, and we published with a number of them, showed promising results, I would say, in vitro. And that was accompanied by people doing RNAi. At that point, it was before CRISPR was invented, showing similar potential efficacy of reducing function of the PAC proteins. But one lingering issue, which is the main thing I want to talk about, is there was more than one PAC isoform. And so there was a question about whether all of the isoforms were doing the same thing and needed to be inhibited in tandem, or one particular one might have a bigger role than another. And I don't think that's totally settled to this date, but it became an important issue because our models, our genetic models, which we were developing at the same time, showed that although PAC1 was dispensable not only for embryonic development, but for most adult functions, PAC2 was a whole different kettle of fish. Unfortunately, disrupting PAC2 function, even in adult mice, was rapidly lethal in terms of giving you heart failure. And this caused, great consternation at the drug companies developing inhibitors, most of which were not specific for isoforms. So Genentech, which I held the most hope for, and a very nice inhibitor that they developed, terminated the whole program. They said, we just can't get a therapeutic window here because we can't separate one isoform from another, and if we hit all three of them, we get too much toxicity. So that kind of left things at a standstill in terms of big pharma development, as far as I know. Now, in the meantime, Wade Clapp and I have been collaborating for a long time, and I guess we don't know when to quit, but he had just developed this very nice Nf2 model, which has been discussed earlier today. And so he bred those to our PAC1 minus mice as well as to our PAC2 flox mice. And to our surprise and to our happiness, the PAC2 deletion didn't really affect schwannoma development. It wasn't important, and we didn't really want to target it anyway, but it seemed like deleting it didn't really slow down tumor growth or hearing loss in his mice. But the PAC1 knockout showed some promise in that the mice preserved their hearing and lived longer. And at about that same time, we realized that there was at least one compound out there that Novartis had made and published that was uniquely PAC1 selective. And we've tested in conjunction with some collaborators more than 2,000 compounds against PAC1 and 2. This is the only one out of thousands that shows this kind of selectivity. And it's not trivial. It's about 50-fold selectivity of PAC1 over 2. And so in this paper with Wade that we published earlier this year, that compound, which had never been introduced in vivo before, did show efficacy similar to the PAC1 knockout. Now, it wasn't perfect. It didn't completely prevent tumor growth or hearing loss, but it definitely had a trend. So that caused us to look harder at this compound. Now, Novartis had abandoned it. So we figured, one thing about these kinases, it's true for FAC, that was just discussed the last time, and for PAC, in addition to having kinase function, they have significant scaffolding function. And when you use this compound from Novartis, it hits the kinase, but not anything else, and it showed some promise. So we thought, well, what if we could turn it into a degrader? I mean, that's the thing that people do these days. So we got together with Nathaniel Gray, who's one of the world's experts on degrader technology, and he made a series of compounds based on this Novartis parent, and some of them were pretty good at degrading, not perfect, but they will eliminate 70, 80% of the PAC1 protein. And a couple of interesting things happen, and I'm almost at the end here, but one is by adding this linker, it actually increased the specificity of the parent for PAC1 over 2. I don't know exactly why that is, but instead of being 50-fold selective, it's more like 500-fold. So you gain tremendous selectivity by adding this extra piece to it. And of course, it was degrading PAC1 and not PAC2. So we have put it to the test, and I have to say, we have not put it to the test in NF2 scenario yet, although that's something that's ongoing currently. But we took a number of cell lines that were known to be either PAC1 dependent or PAC2 dependent, and asked, what will this compound do? And the answer is pretty clear. In the PAC2 dependent lines, nothing happens. You can add the drug even to high concentration. Basically, the cells just keep on growing, and as if no drug had been applied. In PAC1 dependent cells, we gain about a log of potency. So now we're using it at low nanomolar doses, and as we watch the target disappear. So I'm going to kind of leave it at that. I mean, this is obviously a scenario where we would like to, and we are testing it in an NF2 setting, but our trend is to try to take this existing drug, and I think this has been done with FAC as well. There's a FAC degrader out there. I don't know if it's been tested in NF2 yet, but for the same general reason, I think this no modality may resurrect some of these old targets. They may be effective even without the degrader, but I think the addition could buy you a lot more potency.
Well, thank you, Jonathan. So Dan, I saw you nodding your head a few times during that discussion. So maybe talk a little bit more about what your experiences have been since you've been working in the space, and then we'll get into another topic a little later.
Yeah, so I'll admit that I'm not in the NF2 field, but my lab has been, working quite heavily, actually, also in collaboration with Novartis on developing chemical approaches around ligand discovery, and we've been applying that to go after drugging undruggable targets in cancer, and we've also been using it to expand the scope of targeted protein degradation, and also to develop new modalities beyond degradation. And so, you mentioned PAC1 and FAC PROTACs. That's certainly something that we haven't been working on those targets, but we've been, really expanding the scope of novel E3 ligase recruiters that we can use to, get better selectivity, better degradation profiles beyond kind of using just 3-1 and VHL ligands. And I should mention, I'm just looking at the kind of pathway maps of all the, important pathways in this disease. And, among these targets are many kind of undruggable pathways, right? Classically undruggable pathways that I think we now actually have the capabilities of directly targeting, if that makes sense, to this disease. So, for example, Merlin, you could, think about developing small molecule stabilizers or reactivators of Merlin function, or you can think about, for example, targeted phosphorylation of Merlin, or, for example, YAP and TEAD. There are now many efforts to go after drugging, those targets directly, or, for example, you can develop a molecular glue stabilizer between 1433 and YAP. That's something that my lab has, been able to successfully do. Or, for example, going after tumor suppressors like LATs and trying to stabilize those with our recent, deubiquitinase targeting Chimera platform, where we can deubiquitinate and stabilize protein targets instead of degrade them with PROTACs. And this is just really the tip of the iceberg, where we can now do, very targeted manipulation of proteins in a way that would give you the right functional therapeutic benefit for that protein. And, all these targets that, outside of kinases, right, that are currently druggable thanks to, many pharma companies and also folks like NAFDRE, outside of kinases, right, a lot of these proteins that remain intractable. But again, with the advances in both covalent chemoproteomic strategies and also DNA encoded libraries and other approaches, this whole concept of undruggability is really rapidly shifting now. And, we can now develop ligands against really, I think, any of these targets, I think, that are in this pathway map that's in your background. And so, now it's a function of which effector proteins do you couple it to, whether it's an E3 ligase and PROTACs to degrade it, or whether it's a DUB or a phosphatase or kinase to do targeted manipulation of these proteins to confer therapeutic benefit.
I think these are very exciting developments. In terms of, especially in terms of stabilization of the repressors. So I think that's really a great opportunity. I'd like to turn to Christina, because she's used a chemical proteomics approach for finding maybe an unanticipated role for PI3K. So maybe, Christina, can you tell us a little bit about what you've been doing? You're on mute.
Sorry.
Yeah.
Well, thanks so much. It's great for the invitation to be on this panel. And I know that Bart and I are going to have good conversations about whether PI3 kinase is even a rational target to be pursuing. But I'll give you two examples of two tales of studies for PI3 kinase. So when we started doing high throughput screening with Sanford Burnham about 10 years ago, and I'm a Schwann cell biologist by training. I worked on Schwann cell signaling pathways leading to differentiation into myelinating cells. And then I ran into Merlin tumor suppressor that was involved in integrin and ERB2 signaling. And then last 10 years, I've been doing translational studies. So we collaborated with Sanford Burnham. We set up a 384 well plate assay, and they ran a LOPAC, Library of Pharmaceutical Active Compounds screen. It's about, I forget how many compounds now. It was about 10 years ago. And the lead druggable target was PI3 kinase. And when I joined the Synodus Consortium, one of the inhibitors that was suggested by the clinicians that were part of that, Jayshree Blakely and Scott Potkin and others, was CUC 907, which is a dual HDAC and PI3 kinase alpha isoform inhibitor. And it went up until the animal studies. It worked in for Schwannom. It made the cut of 40 compounds. It was the top 3 compounds. It failed to have efficacy in the Schwannoma genetic mouse model of Wade's collapse, and in Longsheng's meningioma model. So we kind of dropped it there. But by that time, I was already funded to look at PI3 kinase by NIH. So I continued working on it. And I did 175 compound library screen of PI3 kinase pathway compounds. And the top the top drug ended up being CUDC 907. So we continued studying it together with Christine Din, who's a neurologist at the University of Miami School of Medicine. And we found that in the 2 or 3 different human schwannoma cell models that I developed and the mouse NF2 exon 2 deleted mouse model, that it reduced viability and it promoted caspase cleavage. And Dr. Din tested CUDC in freshly resected vestibular schwannomas from patients and found that CUDC 907 also caused a loss of viability and induction of caspase cleavage at concentrations that were about 1 to 10 nanomolar. So we've been talking to the DoD Clinical Trials Consortium with Matthias Dr. Katihanis and others to see if this isn't worth going into clinical trials, because this drug now is known as fimpinostat, and it is in clinical trials for children with solid brain tumors. So all I can say is, just like the brigatinib study was kind of surprising, that rationally you would have never studied a drug that inhibited ALK receptors, because there are no ALK receptors expressed in meningiomas or schwannomas. But lo and behold, it has these off-target effects that are really beneficial. And part of Synodus, we also studied a number of FAK inhibitors, and none, to my recollection, worked as well as brigatinib. So I really think it's some of the off-target effects, not just hitting focal adhesion kinase. The second story, if I may, has to do with the combination drug study that was really surprising. And this also arose from the Synodus Consortium, where, at least for schwannomas, the top performing combination drugs that came out of that screen that was done at NIH, NCATS with Mark Ferrer, were basically PI3 kinase inhibitors, 3 or 4 of them, paired with other inhibitors like a PAK inhibitor, aurora kinase inhibitors. So we continued this study funded by the Florida Department of Health, and we did our mini combination matrix, and we found an alpha delta specific PI3 kinase inhibitor that synergized really well with a PAK inhibitor, a PF309 inhibitor. So we went right into animal studies to see if the single agents, look at the efficacy of single agents versus combination of the two. And we're using a mouse allograft model where we inject mouse Merlin deficient Schwann cells into the sciatic nerve. Within a week, they have a mass in the sciatic nerve. You start 2 weeks of oral gavage, single or double agents. And we were surprised that we saw, both in the single PI3 kinase inhibitor and the combination, induction of cleaved caspase 3, which we really haven't seen a lot of induction of apoptosis either in the human cells in vitro. So Sylvia's work earlier was really nice when she's actually finding compounds that induce apoptosis. And we found that this alpha delta PI3 kinase inhibitor induced caspase cleavage alone and in the combination. So I'm surprised, and I've become quite a fan of the unbiased approach where you just design good experiments, well-controlled. If it's reproducible, you just keep following the science and let the biology lead you to its conclusion.
So Bart, are you surprised by the results, and what's going on?
Yeah, hi. So first of all, thanks for the invitation, and I'm not an NF 1 or 2 expert. So I've been in PI3 kinase for over 20 years, and I had the pleasure to clone one of the PI3 kinase that moved from cloning mice, drug development, approved drugs, but it's more in the immune inflammation leukemia area, not in this area. And Christina, this is really interesting data, and it sort of echoes what we have seen over many years. PI3 kinase, I think in every cancer, it's there. If you look at the Merlin pathway behind, on Richard's, it's to the left, to the right, a cell will not proliferate without having PI3 kinase on. So it's always there. And so there's really then when people found that PI3 kinase pathway is also mutated in cancer, that raised all those expectations. Okay, PI3 kinase inhibitors are going to cure every cancer, basically. Easy. And the early PI3 kinase inhibitors were actually quite cytotoxic, and I think they have misled the field because, and proposed as a cure for every cancer, really. And when we then went with cleaner inhibitors or cleaner, isoform-specific mice and all that sort of thing, PI3 kinase is not really controlling cell death directly. It's a metabolic pathway. It switches off the metabolism. The cell goes into kind of a hibernation state, but you don't kill it. Okay. So I'm surprised to hear about apoptosis. Over 20 years, every paper I submitted, referees wanted to see apoptosis. I could not prove it at PI3 kinase selective doses. So for me, it's difficult to understand where the apoptosis comes from. I think that's maybe answering. I have a lot of comments and questions in a way, but maybe if I have a question to start with, Christina, when you say you can actually see apoptosis and caspase cleavage, but when you actually look at how many cells you effectively kill, it's maybe 5% out of the population. So the caspase cleavage is there, but it's a sort of a knock-on effect from maybe working on vasculature, the cells really getting exhausted. It's very difficult also from a signaling perspective. PI3 kinase is not controlling, to a large extent, bad or cytotoxic pathways. It's not doing that. So the apoptosis, if it's there, could be a knock-on effect. Or using the GDC or 941, the delta alpha inhibitor at too high concentrations where it starts to hit other kinases than, PI3 kinases. So how much apoptosis do you see?
I do not recall the percentage, but in a field, you might find 10 or so cells that are expressing cleaved caspase. It could very well be an effect on either the metabolism of the cell. It could be an effect on, we were discussing earlier, perhaps on activation of or action on immune cells or other cells in the stroma. I'm just reporting what we find.
Sure. And do you see regression or stabilization? Do you see do you see regression or stabilization?
Oh, no, we see, well, our assay in our animal models is looking at growth rate over 2 weeks. And we basically get nearly a 75% decrease in the fold increase. So it slows down growth significantly.
Exactly. So, if you're looking after the disease stabilizer, PI3 kinase inhibitors could do the trick. That's what I just said. It slows down the metabolism. It sort of brings the cell in a hibernation state. In fact, the same as what was mentioned before, an mTOR inhibitor, we're talking about the same pathway. So, look, I think for a disease condition like the one we're discussing today, disease stabilization might already be something you're after. That's a good thing. But if you're expecting it to, this to be something that's going to be curative, on its own is not going to be the case. And I think, I doubt it. Let's put it like that. I'm not, but that has always, and where the PI3 kinase inhibitors have worked or have been approved, they're always in combination. The PI3 kinase alpha inhibitor in breast cancer is in combination with fulvestrant, an estrogen receptor degrader. In leukemia, the PI3 kinase delta inhibitors, together with chemotherapy or antibodies against CD20 to kill the leukemic cells. So I think in a combination therapy, you can have let's say, a disease impact that is regressive, but what PI3 kinase inhibitors are going to do on their own is most likely, and everywhere we have seen this, is a disease stabilization. So I think it's a reality check. I'm not saying there's no room for it, just get me.
No, absolutely. I think for this disease phenotype, when you have slow-growing tumors that sometimes don't grow at all, the ability to delay any disease progression, I think the patients would be very happy to have a disease stabilizer. That also brings up the point as to how the preclinical scientists should design their endpoints for their assays. Because my understanding is the clinical trials, patients, once they meet criteria, are expected to have a reduction in tumor size by 20%. to stay on the clinical trial or to be counted as a success. So those that have a shrinkage of less than 20% are deemed not, the drug is deemed not efficacious, or if the drug just stays, stabilizes. It's been growing at whatever rate for the last 2 years, and this drug allows it to stabilize, but that isn't counted as a success. So we need to have really good communication with the clinical trial scientists so that the translational scientists can design the right endpoints for their drugs.
Yeah, Richard, if I may make one last point.
Yeah,
Richard,
yeah,
one last point. One last point.
Yeah.
Yeah. I think, you're using GDC0941, which is a Genentech alpha delta inhibitor. Genentech had to stop it because it had, it was not tolerable. So my advice here would be, your research, I think I would focus it on PI3 kinase inhibitors that have potential, have been approved, and there's only a few, or that have potential to be approved. So I can advise you which ones to use. But I think going after something that, has actually failed in Genentech's hand because, you can't tolerate, I think one just has to move away. I think that's all
I'm not suggesting we go forward with this drug at all. I'm just showing preclinical effects of a drug. I'm not suggesting this drug go forward. I'm just showing, here's an example where we would never have put this drug into an animal because it wasn't effective in cell models. So it begs the question, what are the model systems we should be looking at? Are the mouse models good? Should we just use in vitro human cells? What's the best way? Because I wouldn't have even thought of, now there are delta inhibitors that are actually quite tolerable in patients for some time. They've been on it for years. So if you find a tolerable drug that a NF2 patient can be on for 5 years that stops progression of disease, I think a lot of the patients would be super happy. So for sure, this is just now giving me the opportunity to look at some of these delta inhibitors that are second and third generation PI3 kinase inhibitors that do not have the toxicity of the original ones. And maybe we should look at this target again with fresh drugs.
Okay, thank you very much. In the last minute or so, I'd like to do a couple of things. One is to go back to Dan for a second because we didn't spend much time on the doves, which is a very exciting development, I believe. And then I just like to get a quick wrap up from one sentence wrap up from each of the participants of the discussion. Okay, Dan.
So sorry, what do you want me to talk about?
The dubs. The dubs is a very exciting area for development because it's very hard to find activators or stabilizers.
Sure. Yeah. So I mean, that's just one modality that we've been able to develop around. Instead of targeted protein degradation, we can now go after targeted protein stabilization. But we're now going after, for example, rational molecular glue, stabilizers, degraders, targeted phosphorylation, dephosphorylation, methylation, demethylation. Deacetylation, acetylation, really whatever you want to do, right? And we're just building out an arsenal of modalities that we can then apply to whatever target that we want. And so, I think it looks like there's plenty of kind of druggable nodes like kinases within these within this disease, but I would encourage the field to just take a more radical approach of going after these more kind of undruggable nodes, right? Like Merlin itself, or some of these tumor suppressors, and start using these kind of new chemical biology modalities.
Yeah, thank you. And these new modalities are in fact progressing. I mean, especially in the cancer space and inflammation area too. IRAK inhibitors, IRAK PROTACs are moving forward today. So those compounds are in clinic. So thank you very much, Dan. I'd like to just a quick wrap up from Jonathan.
Right. So I'd like to emphasize what Dan was just saying. I've been impressed with the whole sort of PROTAC and its variant technology. I think it adds a lot to what we have to offer. That said, it also opens up some new targets. I don't think Merlin itself is a particularly good one, even for stabilization, because most mutations result in either a very truncated form or a total absence, so that won't help there. But there are some cases it may help. And I think, though, TEAD inhibitors with a PROTAC on it, which I've not seen yet. There's a lot of companies with pretty effective TEAD inhibitors now. I'd love to see or try out one that was, had this addition. And then you brought up stabilizing kinases like LATs or MIST. I think that's a new opportunity. That would be, no one's really tried that because there hasn't been a way to do it. But if these dub-tacs really are a thing, that would be an incredible opportunity to see if we can stomp on the tumor suppressor pathway and, cause apoptosis or loss of proliferation. So I would be very curious about how that technology develops.
Terrific. Bart, any thoughts?
I think I just would like to recap. We mentioned alpha, delta inhibitors. I would go for alpha inhibitors. I think that's all I want to say, Christina. I think the delta inhibitors, that's more immunotherapy. I'm not entirely sure how the field reacts to that. I think the alpha inhibitors in your case are maybe the ones to go for.
Okay, Christina.
Okay, thanks. I want to thank Children's Tumor Foundation and Ed for developing the Synodus concept, where the preclinical scientist, where the basic scientists, translational scientists, and the clinicians all work together, because that really led to a very rapid progression in what we were doing, and allows the communication to a platform for communicating everything that's going on with patients and clinics with the scientists. So I think it's a great format, and I think it's one that needs to be in some way reintroduced or made available.
All right, thank you all for participating in this panel discussion. Really appreciate it. Thanks, Children's Tumor Foundation. Thank you.
You're on the
Thanks, Rich, for this wonderful panel. Thanks, Bart, for coming in as a non-NF2 specialist, but a PI3 kinase expert. And it really makes me think, shouldn't we start consulting more target experts to really look at the field that we are developing?